So it's really a great privilege to have the opportunity to speak to you about some of the really exciting recent work over the last few years that has changed our approach to personalizing cancer treatment with targeted drugs. And you just heard from Bert about some themes that I'll try to expand on a little bit. And I want to start with what I think are two of the key challenges to developing effective cancer drugs. One of these is matching patients with the right treatments. We know there are a lot of cancer drugs in use, a lot of investigational cancer drugs, some of them showing impressive activity, but in very limited contexts. And we need to do much better at predicting who will respond to treatments and who won't respond to treatments. And as you just heard, overcoming drug resistance is yet another significant challenge for us. Here we're looking at the progression of disease in a medulloblastoma patient who initially responded to treatment with a drug called vismodegib. This patient, you can see these blackened lesions here in the torso are representative of metastasis. These are very effectively treated after 2 months with drug, and then a mere 1 month later, you can see these same lesions recur. And as you heard, we still are reliant heavily on standard chemotherapy drugs, many of which have been around for 40 or 50 years. These are cell poisons, in fact, which target cancers by virtue of their ability to disrupt various aspects of the cell division process, which we know is quite active in tumors. And as a consequence, however, they tend to target normal tissues, which also are comprised of dividing cells. And consequently, these can be quite toxic. And unfortunately, we're not very good at predicting how each patient will respond to these drugs. We may have a group of patients with the same disease, for example, a lung cancer, but when we administer these treatments, we may find out that some of these patients only experience the toxicities and no benefit. Others may experience a mix of both toxicity and some benefit. But we need to do better at figuring out who will respond and being able to predict that. So what have we done to improve the treatment paradigm for cancer patients? Well, one thing we've done is to try to elucidate the circuitry of cancer cells that drives their unrestrained proliferation. So this is, unfortunately, a much oversimplified view of a cancer cell, or even a normal cell, where we have the cell surface here, the plasma membrane, and we have a number of proteins, some of which sit on the surface, and some of which exist in the form of protein interaction networks that receive signals via these receptors on the cell surface, transduce these signals in very complex ways that ultimately lead to triggers of cell division and cell survival. And on normal cells, this process is very stringently controlled and works as designed. But unfortunately, in cancer cells, something goes awry by virtue of these mutations that Bert described for you, and this leads to an inappropriate activation of these signals, even in the absence of extracellular cues that would normally be present. And these cells are then dividing uncontrollably. The second approach that we've taken to understanding how to do better with treatment is to elucidate the genomic landscape of human cancer, which you also just heard about. In sequencing facilities like the one shown on the left here, machines like this crank out enormous amounts of genomic sequence data corresponding to patient tumors. That can be displayed in these complex schematic forms that describe essentially the profile of a cancer genome in individual patients, showing how they relate to each other and how they differ. Collectively, by integrating these two types of information, this has really provided some valuable insights into how we should move treatment paradigms forward. First of all, what we've learned is that every patient's tumor has essentially a unique genomic fingerprint. This sounds like a very complicated and messy situation, and to a large degree it is. However, as you heard, there are recurrent genetic alterations, mutations, that are common to some tumors, and this has allowed us to define vulnerabilities in key nodes within the altered cancer cell. Many of these recurrent mutations are in genes that encode a class of proteins called kinases. Many of the proteins shown here are kinases. These play key roles within the cell circuitry that respond to these triggers from the extracellular environment and stimulate proliferation. Oops. So how do these mutated kinases contribute to cancer? So now I've boiled down that complicated cellular wiring diagram to a few key proteins that have been widely implicated in a variety of cancers. They again include some of these receptors that sit on the cell membrane and receive signals from the extracellular environment, transmitting these signals that ultimately impinge on the cell proliferation and survival potential. Now what happens when a gene becomes mutated and it encodes one of these kinases, and here I've used EGFR, a cell surface receptor, as an example. This can become activated by mutation and transmit signals, ultimately giving rise to tumors. So if we zoom in a little bit on this EGFR protein, here it is on the cell surface here. It has a portion that sits inside the cell. This is where the mutations occur. And these mutations, which are found in about 10 to 30% of non-small cell lung cancers, this is the most common form of lung cancer and accounts for the leading number of cancer deaths in the United States. This is mostly seen, interestingly, in non-smokers, which is the minority of lung cancer patients. And this activation by mutation causes EGFR to become activated all the time and to continuously promote cell division. Now, the way this can play out as a therapeutic opportunity is that if we have inhibitors that target the EGFR protein specifically, drugs that specifically block its function, and we can use those effectively in patients whose tumors harbor these EGFR mutations. And we see here as an example in this CT scan of the lung of a patient with a large lung tumor shown here before treatment, and after 2 months of treatment with this drug erlotinib, which targets specifically the EGFR protein, you can see in this patient with an EGFR mutation a remarkable shrinkage of the tumor. And worth noting that a drug such as erlotinib, a targeted agent here, can be taken as an orally administered drug. It's a pill taken once a day, perhaps, and this is by comparison with the much more toxic chemotherapies, many of which are administered intravenously in the hospital setting. And so this is really a new paradigm, and these treatments notably are associated with significantly less side effects in many cases. Now, fortunately, we can model this kind of response linked to a specific genomic feature of a tumor cell in the laboratory. We refer to this phenomenon as oncogene addiction, a strict dependency on a particularly activated mutation in a cancer for the cell's survival. So here we've taken cells from a tumor of a patient that had a normal copy of the EGFR gene. We can grow these cells indefinitely in culture. If we treat these cells for 5 days with this inhibitor drug, erlotinib, nothing really happens. But if we take cells with a mutant EGFR, again lung cancer cells, treated for 5 days with erlotinib, we can see that we eliminate the vast majority of the treated cells. So we've taken this same approach to expand this paradigm more broadly to use tumor cell-derived cell line panels to identify drug-sensitive subsets. The goal here is to generate large panels of cell lines derived from tumors of all types, propagate those in the laboratory, characterize their genomic features as a way to define the genomic landscape of the actual patient population that we're treating. So these are models of the patient population. We then treat them with various drugs or candidate drugs that we think should interfere with the cancer process. We can generate large sets of data that describe the interaction of these drugs with these cell lines. And by integrating that information and through the use of computational analysis, looking at the genomic features of these cells, the response to these various drugs, we can then develop hypotheses about which genomic feature within a tumor type might respond to a particular matched drug, and we can try to validate that clinically. Here's an example where we can successfully use that to highlight the importance of so-called personalization of cancer treatment in the lung cancer setting. In this case, we've profiled 130 lung cancer cell lines, and what we find is, although to a pathologist looking at the tissues from which these cells derive, these may all look the same. In fact, as we look at their genomic profiles, we find that there are subsets defined by specific activation mutations in kinases, shown here, and associated sensitivities to a variety of drugs or investigational agents, shown here. We can also take this approach to find some interesting relationships among unrelated diseases to certain drugs in terms of their sensitivity. So in this case, we've profiled sensitivity to 700 cancer cell lines for sensitivity to an inhibitor of something called the ALK kinase. What we find, as shown in this pie chart here, is that a very small fraction of this 700 cell line panel are actually very sensitive to treatment with this ALK inhibitor. And interestingly, they correspond to very different tumor types. In one case, non-small cell lung cancer, anaplastic large cell lymphoma, and neuroblastoma. 3 very different diseases. But it turns out that all of the sensitive cells, regardless of the tissue from which they were derived, share one feature. That is, they all harbor activating mutations of the ALK kinase, suggesting that if we want to treat effectively the patients that harbor this specific lesion, regardless of which disease type they have, an ALK kinase inhibitor might be effective. And indeed, this has been demonstrated clinically in the case of ALK-mutated lung tumors. So first of all, up here, what we're looking at is a remarkable response to treatment with a drug called crizotinib, which selectively targets the ALK kinase. The pre-treatment CT image shows a large lung mass here, which is virtually cleared by 8 weeks of treatment with this drug crizotinib. What we're looking at here is what we call a waterfall plot. Each of the vertical bars here describes a single patient, and this is the degree of their response to treatment with this drug crizotinib. These are all ALK-mutated lung cancer patients. This is how they were selected for treatment. And here we're looking at the change in their tumor size upon treatment. And you can see that the vast majority of patients show evidence of tumor shrinkage. A small number of patients, however, do show growth on therapy. However, there's a remarkable relationship between the presence of this mutation and response to this agent. So this notion of personalizing cancer treatment based on genomic features that are well defined and associated with an understood biology has really accelerated in a remarkable way the timeline for developing targeted drugs. If we look historically at one of the first examples of personalized cancer medicine, a drug called Gleevec, which also targets an activated kinase found in chronic myelogenous leukemia. The lesion that causes this disease was actually discovered in 1960, but it's only been recently that the drug has come to practice in clinical use in these patients. However, if we look at the case of ALK that I just described, we're looking at a much shorter timeline from the time we identify this lesion in patients until the time we're able to develop a clinical study in which we could show efficacy of an ALK-targeted drug and ultimately lead to a clinical approval. So when we look at the landscape of some of the more highly recurrent mutations that drive cancer through the activation of kinases, we find that in all cases, this is actively being explored, and there are now FDA approved drugs targeting many of these kinases, and there are a number of other drugs for kinases that are currently undergoing clinical evaluation. So this notion of oncogene addiction has really paved the road to personalized cancer medicine where we can match individual patients and their genomic features with specific rationally based therapies. So when we think about the future, what we're really talking about is a scenario wherein a genomic profile of an individual's cancer represented here in terms of its genomic features that may be relevant to treatment, this is the prescription that will guide treatment decisions going forward. We're not there yet, but we're on our way. Now, you've already seen this image, so you know what's coming next, but we obviously face the challenge with any of these targeted agents that they're not curative. They're invariably associated with the acquisition of resistance, and unfortunately, this happens relatively quickly, as you've already seen. Here's an example in the context of the EGFR mutant lung cancer that I described. In this case, we're looking at a pre-treatment X-ray showing a large lung mass here, which is treated very effectively with 2 months of erlotinib therapy. However, 9 months later, this lesion has recurred. Now, when we think about how resistance developed, there are some principles that I think broadly apply, and then a variety of mechanisms at the molecular level. Broadly speaking, what we understand well is that tumors, which of course are comprised of a large number of cells, have cells within them that are not all the same. And clones emerge over time. They harbor different mutations. Some of those might confer drug-resistant properties. And so even if we're successful at treating the vast majority of cells in this tumor with a drug, there will always be cells that resist therapy, and these cells can then go on to repopulate a tumor at relapse. Now, one helpful bit of information is that we've learned that we can model this process to a large degree in culture using these cell lines that we derive from tumors. So here's an example of an EGFR mutant lung cancer cell that's been treated with erlotinib for a month. What we find is that we can select out drug-resistant clones just as we see happening in the clinic. This is illustrated here where we take these cells in culture. Again, we treat them with erlotinib. We can see cells disappear rapidly. They're being killed. And if we wait a little bit longer, we start to see clones of cells growing happily in the presence of drug. So this seems to model fairly accurately what's happening in the clinic. And by using these cells as a model system to understand the mechanisms of resistance, we've learned a few things that turn out to be clinically relevant. So when we focus on the EGFR model, and we see in lung cancers that if we inhibit EGFR with erlotinib, this will lead to the downstream suppression of signaling, and cancer cells will ultimately die. We've identified one resistance mechanism that seems to be relevant in about half of the patients who are treated successfully, at least initially, with erlotinib therapy, and then whose disease progresses due to resistance. They all harbor a mutation, a secondary mutation in EGFR called T790M, which essentially changes the structure of the EGFR protein so that the erlotinib drug no longer fits in there and inhibits signaling. These cells, therefore, thrive in the face of drug. Another mechanism of resistance we refer to as addiction switching, where now another receptor on the surface that's quite capable of sending signals to these same important downstream players that normally control cell division. This gene now becomes amplified and increased in number. So there is lots of signaling potential from this MET protein here that sits on the surface, which can essentially circumvent the effects of inhibiting the EGFR and driving the same end result. In this case, MET is a third example. MET is still playing a role, but not because it was increased in number, but because now we start to see an accumulation in the extracellular environment of a protein called HGF, which stimulates the MET protein to become active. So too much HGF now drives this downstream signaling, and again, the cell doesn't care that we've cut off its EGFR dependency. Now it has a new dependency. Where does that HGF come from? Well, when we grow cells in a dish in culture and we treat them, we're just dealing with pure tumor cells. But in the actual tumor, things are much more complex. The tumor cells are in a microenvironment that consists of blood vessel-derived cells. We have immune infiltrating cells, and other cell types such as macrophages. So can we defeat these resistance mechanisms? Well, we're making some progress on defeating resistance mechanism 1. That's this secondary mutation in the T790M site of EGFR that prevents drug binding by developing new inhibitors, new drugs, whose shape is slightly different, and now they can come in and inhibit EGFR even when this T790M change takes place. And therefore, this drug has the potential to overcome the resistance associated with a T790M mutation. For the other two mechanisms that involve a gain of function of this MET protein, we now come in with another drug that inhibits MET, crizotinib, which I mentioned as an ALK inhibitor. It turns out it also inhibits MET. And now, by combining treatment between erlotinib and crizotinib, we can take care of EGFR and MET should it arise as a resistance mechanism. So I want to talk about, just in the last couple minutes, a couple of the significant challenges we face going forward. One of these is that while I've made it sound quite simple in some respects, in that if we simply identify a specific genomic lesion in a kinase in a patient's tumor, that patient exhibits a nice response. However, what we find in reality, as shown in these two examples here, is that when we look at these response profiles across patients, either in response to crizotinib in the case of ALK-mutated lung cancer, or the BRAF inhibitor vemurafenib in BRAF-mutated melanomas, there's actually a wide range of response. These patients at this end are responding very nicely to drug. Patients at this end are not responding very well. And then there's everything in between. So while it sounds nice and clean, one biomarker that predicts for sensitivity, it's not that simple. And there are obviously other factors that contribute to the patient's response. And so far, we don't really have enough of an understanding of what those other features are that we might want to interrogate in order to predict more accurately the nature of response. Another big challenge that you heard about already is intra-tumor heterogeneity. When we biopsy a tumor, we're actually looking at a very small fraction of the total tumor burden in a patient. We're looking at a snapshot that is probably not a very representative picture of what's going on throughout the patient's disease. In addition, what's going on in the primary tumor may be quite different from what's going on in the metastases, which we rarely biopsy. So even though we can look at, and if we do this systematically, what we find is that when we look at the mutational profile, it's actually quite different depending on which part of the tumor we look at and whether we look at the primary tumor or the metastasis. That obviously complicates our ability to accurately predict what the appropriate treatment should be. So just to summarize, I've described for you how personalized, what we call rationally targeted therapy with cancer drugs has significantly impacted clinical outcomes. However, rapidly acquired drug resistance remains a major impediment to achieving cures. We can model this drug resistance in the laboratory, and that's revealed multiple distinct mechanisms and has already informed therapeutic strategies to overcome at least some forms of drug resistance. Going forward, there are some key challenges. First of all, we still have a limited understanding of the tumor genome. We can map it, we can describe it, but most of what we see in the tumor genome we don't fully understand, and we don't understand yet how to translate that into predictions about treatments. We also have the problem I just described of intra-tumor heterogeneity. Given how we currently biopsy and diagnose cancer, this will be a problem going forward. And then, as we understand the importance of combining drugs to deal with problems like drug resistance and to target these multiple nodes of cancer that are clearly driving disease progression, each one is associated with its own degree of toxicity, and as we start piling these on, the toxicities will only mount. And then lastly, I want to mention that there are practical limits to how we can stratify patients for treatment and how we can go about drug discovery. For example, is it economically viable to spend $500 million or so to discover and develop a new drug for a patient whose tumor is a 1 in 1,000. We have a good reason to believe that that patient would respond to a treatment against gene X, but it's simply too costly to develop a truly personalized medicine for a 1 in 1,000 event. And similarly, can we even diagnose a 1 in 1,000 event? Is it worthwhile to develop a test to detect that 1 in 1,000 event if we don't have a drug to match? So these are some of our challenges going forward, but despite that, I think we're really seeing an important paradigm change in our approach to personalizing treatment, and these new targeted agents that are being developed as a as a consequence of our understanding of the wiring of the cancer cell has really moved treatment strategies forward, and I think is already having a significant impact on outcomes for patients. So thanks for your attention.